Alrighty, thanks everyone for joining us today. It is my pleasure to introduce Khanh Dao Duc. Professor Khanh is working in the Department of Mathematics at University of British Columbia. His research combines mathematical, computational, and statistical tools to study fundamental biological processes. So today he will share with us some of the related work about translation dynamics across scales and systems.
Okay, thank you for the introduction, Ying, and thank you for coming to this talk. Talking to you right now from the campus of UBC in Vancouver, and if you guys have never been here, I just put a picture of the campus on the bottom left. You can see it's really quite beautiful. You can see the ocean and the mountains. That's the view I have. Every day I can come to work, and pointing, with the red arrow, this is my office in the math department. And it's an opportunity also for me to just acknowledge that this campus is located on the traditional, ancestral, and unceded territory of the Muslim people. So today I will talk to you about the research I'm doing at UBC with my group. I'm very happy to have the opportunity to talk to you today for this Math Bio seminar. We also have a very strong group of people in Math Bio at UBC doing all kinds of research. And what I'm doing more specifically is focusing on translation dynamics. So I'm working at the molecular and cellular scale and trying to understand gene expression and especially the determinants of translation. So a big part of this work also focuses on the object that you can see in the background, which is called the ribosome. So you can see the structure of this complex machine, and I'll tell you more about this during my talk. So just to provide some general context, I want just to tell you what translation is about and some basics on the ribosome. So on the bottom here in this cartoon, you can see a cartoonish representation of the process of translating a messenger RNA into a sequence of amino acids that will become the protein. So suppose you have a gene that is encoded as this messenger RNA. The way translation works is that you have these two subunits of the ribosome, and you can see these two subunits here represented in yellow and blue. They would get assembled at the starting site. So it's encoded by the codon AUG. And then after the translation gets initiated, basically ribosomes decode the sequence of codons into amino acids. So that's why this process is called translation. You have 61 different codons that can be translated into 20 amino acids. So there's some redundancy in the code. And you can see it as a traffic process where basically the ribosome gets translocated from the site to the next. So this type is called the elongation. If there are some ribosome in front, there can be some interference possibly. And this is how the ribosome move until they reach the stop codon, where basically you have a fully formed protein. So this is the termination step where the ribosome gets disassembled and basically the cycle can start again. So translation is basically mediated by the traffic of these ribosomes, and they decode the mRNA codon sequence into a polypeptide chain. And this is a process that can be very heterogeneous. The sequence, there can be variation in the sequences, there can be variation in structure that can determine different rates of translation. So I also want to use this introduction of the context to really tell you more about the ribosome itself. And you can see here a picture of the electron micrograph from George-Emile Palade, who actually discovered the ribosomes in the 50s. And so at the time he was doing this electron micrograph of the endoplasmic reticulum. And at the time he noticed these dots that you can see here. And this is why also this is part of the ER, the endoplasmic reticulum, that is called the rough ER. And at the time, he hypothesized that these particles here, these dots, were associated with translation. And they were, he showed that they were made of ribonucleic protein. And so that's what it's called the ribosome. And I really find amazing that nowadays, like 60 years later, using another technique that I will talk about called cryo-electron microscopy, now you can get a fully detailed picture of these particles here at an atomic scale. So like you can, this is what you can see here on the right, full structure of the ribosome with the two subunits. And what you see in green here is called the transfer RNA, the tRNA, that is basically carrying the amino acid that is added to the polypeptide chain. Okay, so I will just give you an overview of my talk today, and just to say that, again, I'm interested in the dynamics of protein translation. And I will show you how, for the past few years, I've been combining different types of data, from deep sequencing, but also images, to mathematical, computational, and statistical tools to understand the dynamics of translation. Okay, so more precisely, I will present a mathematical model that allows to study the regulation of this translation dynamics at the scale of a single mRNA. And I combat that with a technology called Riboseq. And in second part, I will tell you more about what you can say about, tell you more about the structure of the ribosome, the 3D structure, and how it impacts translation. All right, so some motivation first. I want to introduce you this technology that I mentioned, Riboseq. And so it's a deep sequencing technique that allows to isolate ribosome protected mRNA fragments. So suppose you want to study a gene. So there are around 20,000 in humans, 6,000 in yeast. So you want to know, you want to understand the dynamics of translation for this specific gene. So what you can do is using this technique, which is high throughput. So it means that you can study simultaneously thousands of genes. And what you would do is look at the content of a cell, let's say, and look at all the mRNA that accounts for a specific gene. And these genes that are, these mRNAs that are getting translated, they are occupied by ribosomes. So they form these polysomes, and the technique of Riboseq consists of digesting all the content that is not protected by these ribosomes, and then mapping the fragment back to the genome. You can get a kind of snapshot or picture of the density of ribosomes along a coding mRNA sequence. So you get these profiles here, these plots that we call ribosome profiles. And so I can, here at the bottom, you get an examples of two such profiles for two genes in yeast, where you can already see the kind of heterogeneity in the signal. So basically, you can get the intuition that this density tells you something about the translation dynamics. So if you had, for example, a posing site, you would observe an increase in density and vice versa. But what I was looking for when I started thinking about this was really an exact kind of quantification of this process, and trying to understand more what we can get from these profiles.
Sorry, can I ask a quick question?
Yeah,
Is
sure,
this
go ahead.
from a single cell?
No, usually you do it in bulk. You don't, that wouldn't be enough, I think, depth if you do it with a single cell.
I see.
So you do it in bulk. You form a lysate, so you make everything well mixed. But it's still, I think, a quite faithful representation of translation dynamics if you just get same types, for example. Yeah. You can also do something a bit more specific. You can do some targeting also to look at, let's say, genes that are associated with a specific region of the cell, like the ER. But I don't think you reach the single cell resolution. Okay, so this is actually what I
also had a question.
Oh, go ahead.
Sorry. So like the dynamics of this is it like they all start at a start codon, but you can like randomly add more ribosomes and then they'll proceed down the sequence at different rates or something like that, or maybe they shock or something.
Exactly. Yeah. So I'm going to tell you a bit more about the modeling of this process, but this is basically you basically describe you basically describe the whole process.
Okay.
So I also want to tell you a bit more aside from the technique, the applications and what it can be used for. So you can really use it to study translation, so compare genes together. And this is what a recent colleague of mine did at UBC, Ethan Greenblatt, who started basically a year after I arrived, and we really started collaborating here at UBC, and it was a very nice just convergence of work, I think. So I started working on translation dynamics when I was doing my postdoc at at Berkeley. Ethan was doing his postdoc at the Carnegie Institute, and he was looking at ribosome profiling, and he was interested in the effect of a specific gene that is associated with autism. And it was shown 10 years ago now that this gene had an impact on translation in the coding region. And basically the protein associated with this gene was targeting some specific locations and would basically induce some increase of ribosome densities. And so 10 years ago, they did this experiment doing this ribosome runoff where they just stop translation and they can just isolate genes that have more ribosomes. And they found that this protein would impact the density of ribosomes. And so what he did was using ribosome profiling to get a more precise picture of it, and found out that there was an impact on translation, what we call translation efficiency, due to this gene. And what you can see here in the bottom is a comparison between the ribosome profiles. You can see here how the density was changing when you basically knock out this FMR1 or FMRP gene. But there's still some debate in the community to know if this effect would basically impact the ribosome, the production rate, if the production rate of a protein would be then limited by the initiation step or the elongation step. Okay, so I'll be, I hope that at the end of this first part, this will be more clear. But right now, what I just want to say is that when Ethan came to me, we are discussing, okay, can we connect the Riboseq data with some mathematical model to understand this question and to answer this question? Okay, so I'll tell you more now about the mathematical model and what I started doing when I started working on this project. So it was in 2014-15, and so I was thinking of modeling ribosome traffic and connecting that to this deep sequencing data. So it's actually something that people have studied before, and for that they introduced this model, which is called the totally asymmetric simple exclusion process. And I'm giving here the most elementary version of it. You can consider a continuous time Markov chain that can model the occupancy of your particles along a lattice. So you give yourself a lattice, which is, let's say, your mRNA sequence. And these red particles here can be considered as ribosomes, and they can basically bind. This is the initiation process with the rate alpha. They can translocate along the grid, and if there's a particle in front, you don't move. Okay, so you can consider that all these moves to go with exponential rates. And at the end of your lattice, you can exit, and the rate for this is given by this parameter, let's say beta. So this is a process that was introduced actually by McDonald et al. In biopolymers in 1968 to really model translation. And it was more formalized in the 70s. And then there were a lot of research on stochastic processes on this process. So I won't go into the details of what people have done, but there's really a lot of work. I just want to mention that this is a process that has been used in a lot of biological applications and contexts, even outside of translation for traffic systems and biophysical transport in general. Okay, so this is a pretty simple process, and what you can get from there is the following. So in the 90s, there was this statistical physicist, Bernard Derrida, who used this matrix ansatz to quantify 3 different regimes of transport. And so what is very nice with this process is that you can really obtain a phase diagram that can basically describe the different regimes of the process. So I'm putting here the phase diagram. You can see it in with this plot. There are 3 different regions, and they are determined by these 2 parameters, the entry rate and the exit rate, alpha and beta. And so there are 3 different regimes that you can have. You have what we call the low density regime, where the traffic is limited by the entrance or the initiation. And this is basically where when, as I said, there's not a lot of particles coming in. And so you'll basically end up on a road with very few particles. So that's, when I did that, I was at Berkeley. So I just put different pictures of things that you can visit in California. So this is the Death Valley, where you don't have too much vehicles. Conversely, you can imagine what happened when your traffic is limited by what's happened at the end, and you get this high density regime where you have these long queues that form because people are taking too much time at the end to just leave your lattice. Okay, so this is the Half Dome in the Yosemite National Park, and so the view is pretty nice up there. So that's why people are just waiting to get picture there. So you have this high density regime, and then there's a region of your phase diagram where no matter how you increase the entry rate or you increase the exit rate, you're just now limited by the capacity of your particles to move inside your lattice. And so this regime that calls the maximal current regime, and this is when the biological analogy would be that here you're limited by the elongation step. Okay, and so there's some nice phase transitions, and people have also studied this, but I'm not really interested at this what's happening in the critical region. I'm more focusing on the general behavior. Okay, so this is, I think, a pretty good and intuitive representation. But the problem at the time when I started working on that is that I wouldn't have been able to connect this theory directly with translation and with the ribosome. And there are 2 reasons for that. The one reason is that first, in the previous picture, I showed you just particle occupying one site. And in reality, your ribosome is occupying 10 codons. So if you want to realistic model translation, you have to generalize this process with particles of extended size. Let's call it this parameter L, and L is equal to 10 for ribosomes. The second thing is that in the previous picture that I gave, the rates were the same at each location. It was P or 1 if you want to normalize the process. And in reality, as I said, there's a lot of heterogeneity along the sequence. And so you want to work in a framework where your hopping rates are basically inhomogeneous. Okay, so basically what you have to do is to generalize this simple TASEP model to what we call the inhomogeneous L TASEP model. And people have studied different variants of it, but in the most general version, the problem that you have when you look at what we call the stationary master equation that describes, let's say, the average particle density, which is what you get from the ribosome profile. The problem is that you cannot solve this directly because you get these higher order moments that give you a set of infinite equations. Okay, what you could do, for example, is to do a mean field approximation, and that's what people did. But the problem is that they are inaccurate or they lead to numerical instabilities. Okay, so what did we do then? We worked we worked on a specific regime, considering that your lattice can be very long. We basically transform this discrete set of equations into a PDEs. And to do that, you go to the what we call the hydrodynamic limit of the regime. So it's basically like trying to look a process from far away, squinting your eyes, and then rescaling accordingly in time. Okay. And the advantage of doing that is you can turn this the master equation basically into a PDE that can describe the density. Okay, and this is the equation that I've I'm writing here. Okay, so rho is the density, lambda of x is the rate function, so the rate at which your particles are moving along the transcript. And you have an extra term here, a function g of rho, and this is a term that basically accounts for all the interactions between particles. So I think this is an equation that is pretty not too hard to understand. So if you're looking at how the density varies over time, you look at locally at the rate at which your particles are moving. It's proportional also to the density, and because you cannot move if you have particles in front of you, you have this term here. And if you put, for example, L equals 1, you would basically obtain 1 minus rho. So basically, this tells you that the flux of particle at the site is proportional to the rate at which you're moving, the density of particle you have at a specific position, and the probability of not having a particle in front of you. Okay. So you can actually prove this that you get this limit using a mapping of the TASEP to a dual process, and I won't go too much into the details, but this is we prove that in this paper here. And after that, we focused more on the PDE. So it's a first order PDEs that yields a conservation law. And so you can use the method of characteristics to understand the different what you get at steady state. All right, so now keeping the previous picture in mind, what you obtain now is a generalized phase diagram. So it's basically almost the same as what I showed previously, but here you have some changes, and I will just go over them. Basically, your PDE now yields two possible branches of stationary solutions. Okay, and these branches basically are giving you the low and the high density regimes. So if you take the lower branch, this is now you're in the low density regime, and you can characterize the density. And you can get also J is the flux. So it will be the production rate. So the rate at which your protein is produced. And you can see that is determined by alpha, the initiation rate, and some conditions. Lambda naught is basically the rate at which you're moving once you're inside your lattice at the starting site. And vice versa for the high density regime. Now the boundary is changing a bit. It's not a straight line as before, but the main, I think the main novelty is that is in the maximal current regime, where now you would observe some branch switching between the high and the low density regime. And so the picture that you can have is the same as if you were putting a toll on the road. So I'm sure you have all experienced that when you're driving on the highway and there's a toll, you would really have to wait upstream, and then once you pass the toll, then the traffic clears out. So this is basically what this regime here is describing. You're just waiting until you reach the site where you have your elongation rate is minimal, and we call this rate lambda mean. And then once you reach this part, then you switch and move to the low density range. Okay, so I think the take-home message here is that even if you added a lot of different parameters, really make the process inhomogeneous, in the end, the process is still very similar, and you can use a few set of parameters to really describe the different regimes of transport. Okay, I just want now to get back to the original application that I mentioned in this collaboration with Ethan Greenblatt. And this is now we worked on this for a few months with our student Keegan, and now there's a paper that we submitted and is now under some minor revision. So hopefully it will be accepted and published soon. But basically, we use this same idea to try to understand if the impact of this gene, FMRP, can induce a transition in this phase diagram. So basically, you want to know if the action of the gene can lead to a change of limiting step for the translation of the genes that are targeted. And so we basically looked at the reads and compare the profile, compare the different distributions of the genes. And this is not looking at FMRP, but another experiment where we know precisely that by changing, by starving a cell, you can really make this transition between the initiation limited and elongation limited regime. And you can see that comparing this profile, you can really see this phase transition at the level of the profile. And it's a way for us to verify that our method was able to detect that. And so the method is pretty simple. I should have started with that. What we do is basically looking at the CDF associated with the profiles and computing the Kolmogorov-Smirnov distance between these two profiles, and then look at the distribution of these KS statistics in order to detect such a possible transition. Okay, so there's a separate implementation and protocol that actually we just submitted yesterday. A lot of bioinformatics involved just to process the reads, but hopefully the method will be available for practitioners. So we just applied that to FMRP, and our conclusion was that actually, contrary to what some people were saying, although you see a change in the density, there is no transition actually from the LD to the MC regime. So that's what we get. I don't want to go too much into the details because I want to spend some more time on the second part of my talk. But basically, when you do the comparison and using this idea and this now visualization of the ribosome traffic, you can clearly, I think with confidence, say that translation is still limited by initiation, even if FMRP is targeting some specific, when FMRP is targeting some specific genes. All right, so yeah, I want to move to the next, another application that I did, and it will lead me also to look more closely at the ribosome. And so what you can do actually from the theory, now that you have analytical formulas, is that you can really basically solve the inverse problem. Suppose you have a profile. What you can do in principle is really to use the theory of the TACEP now to just infer what were the original rates. Okay, and this is, we implemented this inference procedure to estimate the rates. And this is a comparison here between the simulated and the experimental profiles. So under the TASEF model, we're able to tell basically what are the elongation rates that can lead to a specific profile. And so the nice thing, once you do that, is that you can use it across many genes, so thousands of genes. You can look at all the sequence context, and you can start thinking about what are the determinants of translation. So this is something that I tried to cover extensively in this paper in PLOS Genetics in 2018. And what I want to tell you more here is how we call the exit tunnel interactions impact translation dynamics. So let me tell you more about these exit tunnel interactions. So to do that, we are getting now, let's take a look at what's happening at a single ribosome level. And so you have this picture of translation that I showed previously with the 2 ribosome subunits, the tRNA, and this is basically the protein that gets built. And so when translation happens, actually your protein gets formed and has to go through a compartment of the ribosome that is called the exit tunnel. And what people have shown is that actually you can design specific sequences, for example, to just arrest translation. And due to just the geometric and biophysical constraints of this compartment, there can be some impact on how your chain is able to move, and then a modulation of translation dynamics. So this is an extreme case where you can see an arrest of translation, but what we are thinking of is, can we actually see a more general modulation of these dynamics across all the genes? Okay, so we're looking for some other determinant, but basically upon looking at different determinants of translation dynamics, we're trying to assess if the ribosome tunnel and the interactions between the ribosome tunnel and the protein that's getting translated, if these interactions have an impact on the translation dynamics. So don't want to go too much into the details. This is in the PLOS Genetics paper, but basically I used a linear model to try to fit the deviation from mean codon elongation rates, because the codon identity was one of the main determinants, but there's still some variation. And what we found is that if you focus on what's happening at the early stage of translation, so when your polypeptide is still inside the ribosome tunnel, like in this picture on the left, then if you look at the sequence of amino acids, what we found out is that if you have positively charged amino acids that were translated before, then it directly impacts the translation speed afterwards. And in a way that positively charged amino acids have a positive effect, so they increase the translation speed when they're here, and vice versa. Conversely, negatively charged amino acids will have a decreasing effect on the elongation rate. Okay, and this is just what you can see on the right is just a fit between our model and our observations. Okay, so this was pretty intriguing to us, and I think the fact that these two combined effects had these opposite, basically these opposite effects, it suggested that there would be some influence of the electrostatic potential. So that's the last piece of work that I did in this PLOS Genetic paper, is trying to get the sense of the biophysics of the ribosome exit tunnel and see if the tunnel biophysics is in agreement with the observations that we get from sequencing data. And so you can basically think about the process of having a particle that is inside a tunnel of specific geometry. So this is basically your ribosome tunnel. It's not a pure cylinder. There's some variation. And if you look at the diffusion of a particle, through such confined geometry, what you can do is to simplify this the 3D diffusion into 1D, but then you have to take into account the geometry. So what you do is that you project, you're looking at the diffusion across the longitudinal axis, and the effect of the geometry leads to what we call an entropic force. That basically is saying that if you increase the radius, you increase the entropy, and so the longitudinal flux is decreasing. So this is what Zhang did in 1992, and he studied this this process, and the equation that was associated with it, which is called the Fick-Jacobs equation, where there is basically now two main forces that drive the diffusion of your particle. First one due to the electric field, so the electric potential that I'm calling V, and this geometric entropy that is basically proportional to the area of the log of the cross-section area. And so, long story short, when you look at the structure of the ribosome tunnel, so at the geometry and the electrostatic potential, so you can get the electrostatic potential by just looking at the local charges and compute the Coulombic potential, but this is a very simplified version, but I was just looking at qualitative results in a sense. What we found is that first, the geometry tells you that there's an entropic barrier that forms at early stage. So basically, because your entropy is increasing, it's harder for your chain to go through the tunnel. But this barrier can be compensated if your particle is charged, because then your electric field can point you outward, in the outward direction. And it's the opposite if you have a negative charge. So in a sense, these forces and the direction of them are in agreement with what we observed for the translation speed. Okay, so there's something here that I didn't mention is how I could get these profiles, and I get to that in the final part of my talk. Okay, I just want to conclude here to say that I just gave you a stochastic description of this stochastic model of the ribosome traffic along the RNA. And I think what's very nice, and that's a good use, I think, of a mathematical model, is that this can lead us to quantify the different regimes of transport and have some phase diagram transition where we can really understand the important factors that can govern the process that we're studying. And I try to connect that with this deep sequencing data to study the specific action of a gene, but more generally understand the determinants of translation speed. And this led us to look at things at the atomic scale, where now we are looking at the structure of the ribosome itself. There's some further work I want to mention that has to do with how you can actually use this formula to now visualize all the quantities that are of interest, like density, the flux, and the phase diagram, without using stochastic simulations like the Gillespie algorithm. So that's some work that we published further. And there's also some more principles that we derived from this study, and this is what we did in this paper here in Cell Systems. Okay, I'll say a few words afterwards about the structure and the exit tunnel. But before that, yeah, if you guys have any question on this part, I'm happy to answer it now.
I had a question about slide 14.
Mm-hmm.
So the simulation data, is it from one trial or multiple trials?
So there are no simulations here, actually. So it's, yeah, I went a bit fast here. I'll tell you more about the simulations. Actually, this is the second part of my talk.
Oh, okay, okay. Because I thought some of them were from experiments, some of the data points, and
the data points are from experiments, or the opposite. I can't remember. Maybe the green. I think no, the red is from experiment. And so what you can see in red are basically the variation in local speed when you average over all the genes when you start translation at the early stage. And in green, it's a fit where basically we put all the biophysical parameters of the sequence. So for example, we put all the information on the sequence context. And we are trying to do basically a linear regression to understand what are the important factors that determine translation speed. So you can think about the fact that it has a charge, the hydrophobicity. So it's basically a statistical analysis that I did here. There's no simulations.
Oh, okay, okay.
Yeah.
Sorry.
Yeah, sorry, I went a bit quick on this because it's it's basically some linear regression that we did.
Thank you.
But I'll tell you more about the simulations. I hope I have time. Yeah, okay, let me try to spend 5 minutes to tell you more about this. Yeah, so what I did, you know, at this stage, you know, looking at the tunnel, we were quite excited about it, but we asked the question, okay, how universal is this basically? We did this study in yeast, but the fact that the charges are important and this interplay with the geometry, it could change from species to another one. And it basically, it's like putting an kind of evolutionary lens on it, and also taking advantage of the fact that you have now plenty of structures available for different species and across the different domains of life. So eukaryotes or prokaryotes. So what you can see on the left are the different structures of the ribosome that are available for different species. You have bacteria that are indicated in B, archaeas, and eukaryotes with A and E respectively. So we did the study in yeast. Yeast is in here, but you also have the structure of the ribosome is also available for humans, for E. coli, etc., etc. And so we are thinking, okay, can we understand the geometry of the exit tunnel from an evolutionary point of view? And it's basically like modeling the tunnel as a quantitative trait. And that's what people did actually in math bio. At the early stage of math bio, 100 years ago, people were, for example, looking at how shape would vary between different species. So looking at this phenotypic trait and try to understand the differences between them. Now, I think what is really nice is that with the technology available, we can now use the same idea, but now looking at the molecular scale. And this basically leads to another mathematical question, which is what kind of morphometric to use in order to do this comparison and try to understand differences across species and domains of life. So, okay, this is just a technical detail on how you can extract the tunnel geometry. So if you're more curious, I can tell more about it later on. But basically what we did was to extract the tunnel geometry for all the different species. So similarly as what I did in the PLOS Genetics paper. But now you have basically a family of tunnel radius plots that you can compare. And so what is out of this comparison, you can get a pairwise distance matrix and you can do some clustering. And what we found was that if you do this clustering just based on geometry, you obtain something that is consistent with the phylogeny that you would get from sequence analysis. Okay, so you see here that the trees that you get separates bacteria and eukaryotes with archaea in between in a similar fashion as the phylogenetic tree that you get from sequence analysis. So that really suggests that there's some association between sequence and geometric evolution. And for more, we covered that more extensively in this paper. But there is one specific key feature that I want to mention here that is very important, and that will connect us back to translation. Okay, and the very important feature that we found was that there's one main difference between bacteria on the left and eukaryotes on the right, is that there is this region here that we call the constriction region, where you have first very conserved constriction site. But what happens in eukaryotes is that because this protein UL4 has an extended arm, it creates a second constriction site. And you can see it in the profile here on the right, is that when you look at the profile, you really have this trough that is deeper in eukaryotes. And that's really something that drives the separation between eukaryotes and bacteria, basically. And now, the fact that you have here something that is more constricted, and if you think about translation, intuitively you can think, okay, this may be this may have an important impact on translation dynamics. So we wanted to investigate this question a bit more. And so for that, we use simulations. Simulations basically of the polypeptide chain dynamics inside the tunnel geometry. And so we represent the tunnel with these two constriction sites. In eukaryotes, and in bacteria, you only put one. And you can simulate this and consider this as a first passage, an escape process with you can look at the first passage time to the exit for your polymer and the probability of exiting. It's also possible that your chain gets stuck inside. So I have some videos to show actually, but just for the sake of time, I will go a bit quickly here. But what we did was basically doing these simulations, and we focused more precisely on what we call the small peptides, because if your peptide is long enough, then there's enough entropy outside to really pour your chain out. So you don't have to worry about this. And that's also why in the previous analysis on sequence, the effect that I was describing was only observed at the early stage of translation. But basically what we did is generating these small peptides and look at different lengths. So the polypeptide is still within the tunnel, and try to simulate the escape process. Okay, so we had multiple sequences of fixed length, and then we simulate the escape process in two geometries, one where you only have one constriction site, and another one when you have two constriction sites. And so what we found, so this is a work that is ongoing, but we're basically wrapping this up now, is that there's a real effect, the length has a real effect. So there's some critical length at which in eukaryotes, your polypeptide gets strongly trapped inside the tunnel. And so there's a lower probability of escape. And this also impacts the conditional escape time. And this effect here is less present in prokaryotes. So what I can tell you is that we looked more closely at our stochastic simulations, and we found out that at the 12 mer, so when your polymer is 12 amino acids long, this is really the regime where your polypeptide gets stuck in this region between these two constriction sites. Okay, so you see here this marginal distribution of the C-terminus position of the polymer at the end of the simulation. And so you can see that most of the trajectories are concentrated in this region here of the tunnel. Okay, and you don't observe that when you move to different lengths. Okay. Okay, so I just want to conclude here, just to say about the second part is that we are able to process the tunnel from multiple 3D structures. We found some interesting differences between bacteria and eukaryotes. These differences, we are thinking that they have some functional implications. And what we looked more precisely at was the escape of the small peptides. So that's what's happening when translation is ending. And yeah, there's more that I can tell about it, but I just stop for now. Just want to mention some other works that our lab is doing. We're looking at different determinants of translation at other scales. So this is what you can do with in vitro translation. But looking at these ribosome structures actually also led me to investigate how to develop other methods for cryo-electron microscopy, and we are using some pretty cool other mathematical technique like optimal transport or group theory for geometric deep learning. And I just want to advertise a workshop that I'm organizing on the mathematical and computational challenges in this domain. You can email me if you're interested, if you are familiar with Cryo-EM and want to learn more about it. And this also led me, looking at this ribosome, the tunnel shape, think about shape analysis, more generally speaking, and look at application to cell biology. And in particular, I want to advertise this other workshop at BIRS that will organize it next year. And one of the co-organizers is Ashok Prasad, who actually is a former graduate from Brandeis. So I thought it would be nice to mention his name here. Okay, want some final acknowledgements to my postdoc lab. So Yun-Sung was my advisor at Berkeley. I worked with Jamie Kate, who's a structural biologist also. This work also really benefited from great graduate students at the time. So Dan, who did contributed to the math, Sanjit, who contributed to the bioinformatics analysis. And these are my current collaborators and students at UBC. So Frédéric Poitvin is a structural biologist at Stanford, and we are looking at the problems in Cryo-EM. Ethan Greenblatt is a professor in biochemistry doing ribosome profiling. Simka Srebnik is an expert in this molecular dynamic simulation that I showed in the end. And we're working with this group of students, Artem Kushner, Keegan Flanagan, and Shiki Yu. Okay, so that's the end of my talk. I just stop here. I just want to thank you for your attention and happy to take any questions.
Thank you very much for such an interesting talk. So now feel free to ask questions if you have any.
Yeah.
Thank you, Khanh. That was a very interesting talk. I wanted to just ask if you could say a little bit more about this sort of observation that there's a length at which the polypeptide chain is getting trapped. Is that something that is sort of clear from just the relative sizes of things, or is it important to know the details of the molecular dynamics to understand what's going on?
Yeah, okay. There are a few things I should mention. Yeah, I hope I wasn't too fast in the end. I think here, what we really want to study is the importance of the geometry of the tunnel, right? So the way we model the tunnel is just, with this simple representation. People have come with some more detailed representations, also a more detailed and realistic representation of the polymer. So here we have a quite simple polymer, a coarse-grained polymer. So we are not modeling, all the atoms. We are kind of simplifying this into into just a simple polymer. Maybe you want some more details about the polymer, but you can.
Well, I guess I'm curious about this sort of trapping. What does it mean in the context of the simulation?
The other thing is that, okay, we initiate the polymer inside. So you attach the polymer at the start of the tunnel, then you let the system get into equilibrium, let's say. And then at the starting time of your simulation, you just release your polymer, at the start of the tunnel. We call it it's called the PTC, the polypeptide transferase center region. And then you're just letting you run your MD simulations, and you go over a certain time. And basically the probability of escape is basically given by the probability of reaching the exit after a certain amount of time that is, let's say, long enough. So that's the result that we capture. So once you do that, first you can look at the probability of escape, the conditional exit time, and also in this you can look at where you are at the end of the simulation. And this is what I showed in the end, this conditional distribution of basically where, let's say, your center of mass or the extremities of your polymer are at the end of the simulation. And in this critical length, basically what we observe is that most of your polymer is just concentrated in this, we call this pocket region between the two constriction sites. And it's basically, the basic physical intuition is that, okay, if your polymer is short enough, okay, it's small enough so you can go through, these two constriction sites. If it's long enough, then at some point there's enough entropy to pull things out. But when it's in this critical length where, basically your whole polymer gets within this pocket region, then it's too big to pass the second barrier. And this is basically the problem that you encounter. It's pretty, I think in the end, it's not too, there's nothing too surprising, but I think the fact that we were quantifying, okay, something I can show you that I really like, and I didn't mention that in this talk, during the talk. Okay, I can show you actually a poster that my student Shiki presented at the Biophysical Society meeting. So that's the poster that we presented at the BPS. You can see the screen, right? Mm-hmm. So what I didn't show in the end was this plot here. So these are histograms that show you the distribution of the length of real sequences, these real short peptides. And what you can see when you do this histogram is that you can see this bias. So there is less sequences, and yeah, so it may be a bit small here, but you can see it's around 10 amino acids. So that's what we want to say in the paper that we are wrapping up right now, is that, okay, now we have all the simulations, we show there's effect, and there are some possible evolutionary implications. Now, there's some limitation. The limitation that I want to mention is that this is a tunnel that is just a simplified cylinder. There's no electrostatic potential. So the goal is really to focus on the geometry. So we remove all possible inferences of the electrostatic potential. So it's possible that and actually that's what the first part of the talk was suggesting, is that having charges also can help you to exit from the tunnel, and that would be for a further study, basically.
Oh, you're muted, Ying.
Sorry. Do we have any more questions for Khanh?
Just a quick one for me, I guess. So you mentioned the determinants of translation. I just wonder, is that like a mathematical object, or is that like you mean like what causes a translation, I guess?
Yeah, it's more the translation speed that I'm interested in. So think about this. We have, let's say, in human, you have 20,000 genes. These genes are of different lengths, they have different sequences, the mRNA sequences, and they all go under the same process of having ribosome binding and trafficking along these mRNA sequences. So what ribosome profiling is telling you is that there's a lot of heterogeneity across the different genes. And so let's say the big picture and the big question is to say, okay, how can I explain this heterogeneity? What are parts of the sequences, or what are the biophysical parameters that are governing, or if you want to say, determine the rate of translation? So, genes can be more or less expressed, and it's possible that translation plays a big part in it. And so you want to understand basically if, I don't know, codon identity could be the main driver, for example. And in this case, you would have a simple way of predicting the rate at which a gene can be produced. But I think the picture is a bit more complex than that, and that's what I have been trying to study. Does that make sense to you? Is that clear?
Yeah, I guess somewhat. I mean, I'm not really familiar with biology and gene.
Okay.
But yeah, it does. And I think what I get from your talk, correct me if I'm wrong too, is that so the rate of translation in that kernel, it determines the rate of exit, yeah, and then the rate of exit, you try to study that in terms of like how that related to the evolution of other species.
Yeah. So the different there are different things. So the rate of translation is, just the rate at which, when you have your traffic, the rate at which your particles are getting in, moving in and exiting. So that's one of, let's say, the first part of my talk was trying to model and describe, this process and understand what can determine, what's happening at the start also. The last part that I studied is more once termination happens, there could be some potential issues where if your chain is small. I think if the chain is, if you have a sequence that is long enough, you don't have to worry too much about what's the termination process. There's enough entropy for your chain to just let your polymer, your protein exit, get your ribosome disassembled, and everything goes well. But what we wanted to see is what's happening for smaller proteins. And more precisely, trying to understand if I'm having something, a ribosome in yeast or in bacteria, the process could be different. I mean, the geometric differences could lead to some more constraints on how to make translation work efficiently. So yeah. Hope that answers your question.
I see. Yeah. Thank you. Really nice talk, Khanh. Appreciate it.
Thank you.
Thank you.
Thank you.
Any more questions for our speaker? Alrighty then, let's just wrap up here for today. I'd like to thank our speaker Khanh again and everyone for your participation. So we will take another 2 weeks breaking and then see you again by the end of this month. Okay, so have a nice day, everyone, and hope to see you all next time.
All right, thanks again, and see you guys.
Thank you, Khanh. Thank you.
Thank you.
Thanks.